how this plant tolerate the extreme condition temperature is by increasing the percentage of the unsaturated phospholipid in autumn. Okay, so this keeps the membrane from solidifying during the winter, during the cool temperature. Okay. Okay, another example given here is the fish. Yeah, they are capable of adapting to cold environments by changing the proportion of unsaturated fatty acid in their membranes in response to the lowering of the temperature. Okay, another factor that affecting fluidity, yeah, is the cholesterol, right? Okay, in animals, yeah, the cholesterol keeps the membrane fluid. Okay, so it lies. Okay, the cholesterol, it lies alongside the phospholipid in the membrane and tends to dampen the effects of temperature on the membrane. Okay. Okay, right. So the cholesterol has different effects on membrane fluidity at different temperature. Okay, so you can see here, the cholesterol is between the phospholipid, wedged between the phospholipid in the plasma membrane, okay, of the animal cell. So during high temperature or warm temperature at body temperature 37 degrees Celsius, okay, the cholesterol will restrain the movement of phospholipid and the membrane will become less fluid. This is during the warm temperature. Whereas during the cool temperature, it maintains fluidity by preventing tight packing and slow down the solidification of the membrane, right? Okay, so you can see here the cholesterol, the presence of cholesterol within the phospholipid here in the animal cell membrane. So cholesterol also, it reduces the membrane fluidity at moderate temperature, okay, by reducing the phospholipid movement. At low temperature, it hinders solidification by disrupting. Okay, by disrupting it, it will interrupt. It hinders solidification. It inhibit solidifications, or it obstruct solidification by interrupt the regular packing of the phospholipids. Okay, so the cholesterol also act as a fluidity buffer for the membrane. It resists changes in membrane fluidity caused by changes in temperature. Okay, so it functions. Cholesterol functions as a buffer, preventing lower temperatures from inhibiting fluidity and preventing higher temperature from increasing fluidity too much. Okay, so cholesterol extends in both directions. Okay, the range of temperature in which the membrane is appropriately fluid and consequently it function. Okay, functional. Okay, so membrane must be fluid to work properly. Okay, if it is not fluid, then the membrane cannot work properly. Okay, so when a membrane solidifies, its permeability will change. Okay, the enzyme protein in the membrane will become inactive. That's why membrane must be fluid. Okay. All right. So this is the last slide for part 1. Okay. there are some questions. Okay. The first one is list 3 components of plasma membrane. Okay. And then the second question is name 2 types of phospholipid movement. The third one is why is the plasma membrane described as fluid mosaic? Okay. The last question is how unsaturated hydrocarbon chain maintain the membrane fluidity. So students can answer the questions. You can refer to these slides, or you can also get more information from the textbook. Okay, so this is the end of my lecture in part 1. Thank you.
Okay, terima kasih doktor. Okay, students, okay, kita akan berehat seketika dan akan kembali selepas 10 minit. So, jika ada sebarang persoalan, boleh type di ruangan chat Google Meet kita. Okay, boleh take 10 minit dan kita akan kembali semula. Okay, kita kembali. Okay, saya serahkan kembali kepada Profesor Madya, Dr. Norrizah, Jaafar, Sidik untuk part 2 chapter 3. Okay, silakan doktor.
Prof tengah bercakap ke?
Assalamualaikum. Assalamualaikum.
Waalaikumsalam doktor.
Okey, tadi minta maaf. Tadi ada masalah, dia tak keluar bila saya share.
Tak apa doktor, mungkin boleh mula lagi sekali.
Okey, sorry Sorry. Tapi sekarang ni saya dah share. Ada nampak slides?
Belum doktor.
Okay, stop sharing. Okay. Okay. Present now
Boleh doktor, silakan.
Tak keluar lah dia punya slides your entire. Ya, sekejap. Kenapa tak keluar?
Tak apa doktor, kami tunggu.
Okey, lagi sekali sebab tak keluar. Tadi boleh keluar, yang ni cancel. Again. Alright. Share window. Invitations. Kenapa tak keluar? Tak keluar. Tak keluar.
Okey doktor,
tak keluar. No, tak keluar. Sorry. Stop presenting. Tak keluar. Best step. Tak keluar gambar tu. Sekejap. Yes, sorry. I'm so sorry. Cannot present it. Okay, now. I share now. Okay.
Okey doktor.
Alright. Okay, boleh nampak. Okay, I start now. Okay. Now I will continue with the second part of the plasma membrane transport lectures. Okay, so we start with the topics on membrane proteins and their functions. Okay, so here the proteins determines most of the membrane's functions. Okay, if you look at the figures, right? So the proteins, okay, embedded on the bilipid phospholipid bilayer, right? So there are peripheral proteins which bound to the surface of the membrane, and also it has the integral proteins which penetrate the hydrophobic core. Yeah, and if you look here, right, there is also integral proteins that span the membrane, what we call as transmembrane proteins. Yeah, so the transmembrane proteins, if you look at the figure here, it has the hydrophobic parts, yeah, the alpha helical secondary structure. These are the hydrophobic parts. And the hydrophilic segments, which are the non-helical in contact with different aqueous solutions. Yeah? At the extracellular side, these are the extracellular side, and here is the cytoplasmic side of the membrane. Yeah? So these are the transmembrane proteins. Okay, there are 6 major functions of membrane proteins. Yes, if you look here, the membrane proteins act as a transport. It also acts as an enzymatic activity. Okay. And it also involved during the signal transduction. It also involved in the cell-to-cell recognitions, and it also acts as an intercellular joining. It also acts as attachment to the cytoskeleton and extracellular matrix. Yeah, I will go through one by one of the functions, yeah, of these membrane proteins. Okay, first we look at the first function, as a transport protein. Plasma membrane are very selective. Yeah, they only allow certain solids to enter or leave the cell. Okay? So they have the channel here. Channel is like a tunnel where the selective solids or certain solid can move in or out. Okay. So the solids can enter or it can leave the cell through the channels or carriers. Yeah? Carriers composed of the proteins. So if you look at the carriers here, yeah, it involves the ATP. Yeah, the ATP. Unlike the channel pore protein here, okay, it does not involve the ATP. Okay, so the channel pore protein, it provides a hydrophilic, yeah, the membrane protein here, it provides a hydrophilic channel across the membrane that is selective for a particular solute. Whereas the transport carrier protein transport a substance from one side to the other side here by changing its shape. Yeah. So that one is for the channel for the transport or carrier proteins. Now we look at the membrane protein which involved in the enzymatic activity. Okay. So this protein is built into a membrane, maybe an enzyme, right? Can see here there is an active site. Yeah? So the substrate will enter the active site of the enzyme. Okay, and then it will leaving the active site. Okay, so the active site has a very specific size, shape, suited to bind to a particular substrate. Okay, so active site exposed to the substance in the adjacent solution, that it will attach, it will bind to the active site here. Okay, and then another function is involved in signal transduction process. Okay, so signal transduction process, it refers to the movement of signal from outside the cell, resulting in the change of the in a change in the state of the cells. Okay, so you can see here there is a receptor at the membrane proteins. At the membrane protein, the membrane protein here, it acts as a receptor. Okay, so the signal molecule, for example, like hormones, it will bind to this part. It has a binding site here with a specific shape that fits the shape of the chemical messenger or the signaling molecule, which cause the protein to change the shape. Yes, so this allows message to relay to the inside of the cell. Okay, so here it involves the reception, the transduction. Okay, so at the reception, it binds to the receptor, whereas during transduction, it will change the receptor proteins. Initiates process of this change. Initiate process of transduction, usually a pathway of several steps, and then go to the next is the response, where the signal will trigger a specific cellular response. Okay, so these are the process, the reception, the signal. There are three processes here: the reception, the signal transduction, and the response. Okay, so you can see the receptor here, where the hormones will attach to the binding site here, and then it produces a reaction, second messenger, and there is a activation of the cellular responses. Okay, signal triggers a specific cellular response. Okay, the next function is involved in cell recognition. So this a cell's ability to distinguish one type of neighboring cell from another. Okay, so the membrane carbohydrates may be covalently bonded to lipids, the glycolipid, or more commonly to protein glycolipids. Okay, some glycoproteins serve as an identification tags. Right. Okay. The next is cell recognize each other by binding to surface molecule, often a carbohydrate or protein, on plasma membrane. This is important. Sorting of cell into tissues and organ in animal embryo. Okay, basic for rejection of foreign cell by the immune system in vertebrate animals. Carbohydrate on the external side of the plasma membrane vary among species, individual, and even cell types in an individual. So example here given the human blood types. So the blood type A, B, and O here. If you look, the blood types are genetic. There are 4 major blood groups: A, B, and O. Okay, so the blood groups based on the antigen, the cell antigen, which are proteins on the cell surface that are recognized by the person's immune system. Okay. All right. Another functions of membrane protein is as intercellular joining molecule. Okay. So membrane proteins of adjacent cell may hook together in various kinds of junctions. Okay. So there is a gap junctions where it provides cytoplasmic channel between adjacent cells. Tight junction where it preventing leakage of the extracellular fluid. And another function is to attach to the cytoskeleton and extras matrix. Okay, so you can see here, right, in the figure here, attachment to the cytoskeleton and the extracellular matrix. Okay, so microfilament or other elements of the cytoskeleton may be non-covalently bound to membrane proteins. So this one, it helps to maintain the cell shape and also to stabilize the location of certain membranes protein. Okay. So there are altogether there are 6 main functions of the membrane proteins. Okay. So the last slide is the summative questions. The first question is name 2 types of transport protein. And then the second one is briefly describe how membrane protein evolve in signal transduction process. And the third one is name specialized proteins that are embedded in phospholipid bilayer and describe their functions. So this is the last part of, this is the last slides of part 2. Okay.
Okey. Okey, terima kasih Dr. Norrizah. Okey, pelajar sekalian, kita akan berehat seketika. Okey, kita akan kembali selepas 10 minit. Sekiranya ada sebarang persoalan daripada pelajar sama ada di Google Meet atau YouTube Live, boleh type di ruangan chat. Okey, terima kasih. Kita akan kembali lagi 10 minit. Okay guys, kita kembali bersama dengan Prof Madya Dr. Norrizah Sidik. Okay, sebelum kita ke bahagian 3.
Kamera dah belum lah kerja.
Belum lagi? Okay.
Sekejap. Okay dah, kamera okey.
Okay, terima kasih Cik Syam. Okay, sebelum kita ke bahagian 3 bersama Prof Madya Dr. Norrizah Jaafar Sidik, saya akan bacakan 2 soalan daripada pelajar. Okay, yang pertama Wan Muhammad Afif Nazrul Anwar. Okay, Doktor. Dia bertanyakan, Assalamualaikum Doktor, I am Wan Muhammad Afif from S1. I would like to ask a question. Why do we use term segments for hydrophilic and parts hydrophobic? Are they the same?
They are same, eh? They are same actually. The segments hydrophilic and parts, they are the same.
Okay, thank you Doktor. Okay, yang kedua daripada Muhammad Aiman. Okay, Assalamualaikum Doktor. Saya ingin penjelasan tentang how cell recognition involved in rejection by immune system. If there are no compatibility between blood type or of donor and recipient.
Okay, context of blood transfusion, the blood must match compatibility between the donor's blood and the recipient's blood, right? Okay, so this not always mean an identical blood match. Okay, so plasma transfusion, right? The red blood cell from a donor that is type, let's say O, can be transfused into patient of 4 different blood types, right? AB, and O. Okay, so people with type O blood are called universal donors because their donated red blood cells have no A, B, or Rh antigens, right? And can therefore be safely given to people of any blood group. Okay, so people with type AB blood are universal recipients, because they have no antibodies to AB or Rh in their blood and can receive a red blood cell from a donor of any blood type. Okay, so plasma transfusion are meshed to avoid AB antibodies in the transfused plasma that will attack the recipient's red blood cell. So people with type AB blood are universal plasma donors. Their plasma does not contain A or B antibodies and can be transfused safely to all blood types. Okay, so here how cell recognition evolve. So because they have the site of they have the antibodies present and also antigen present, right? So they must match among this the cell, the cell must the antigen must match to the antibodies. Okay, that's all.
Okey, terima kasih Doktor. Okay, sebelum kita ada satu lagi soalan daripada Izrina Izlin. Assalamualaikum Doktor, I'm Izrina. As we know, cholesterol act as a fluidity buffer, but I read some other notes state cholesterol act as a temperature buffer. Is it considered the same?
Yeah, it's also act as a temperature buffer. Yeah, it can consider the same one, right?
Okay, thank you Doktor. Okay, kita boleh sambung ke bahagian 3 atau bahagian terakhir untuk chapter plasma membrane and transport. Okay, silakan Doktor.
Okay, so I will share my slides. maybe it takes time to get it come out.
Okay, Doktor.
It's slow.
It's okay.
It doesn't the slides does not come out. I type the entire script, type the window and also the tab. Okay, I try again. Usually when I just press the tab, it will share. But here, there is no...
They not appear.
It's not appear. Can we wait for a while?
Okay, boleh Doktor.
It doesn't come out. I don't know why. Masih lagi tak keluar lah.
Belum lagi Doktor.
Ya.
Doktor guna PowerPoint tu?
Ya. Saya pergi ke tab tu, dia tak keluar dia punya tu.
PowerPoint ke?
Stop sharing. Yes, PowerPoint.
Uh,
Oh, sorry. Stop sharing. Ada nampak ke? Tak ada.
Belum lagi.
Yeah, cannot share. Window, okay. Okay, sekarang dah keluar. Okay. I will continue the lectures on the plasma membrane, part 3. Yeah?
Silakan.
Okay, I will continue, okay? Okay, I will continue with the lectures on the plasma membrane transport. So this slides, it shows the membrane structure result in selective permeability. So if you look at these figures, right? These are the phospholipid bilayer, right? So here shows the hydrophobic molecules such as the oxygen, carbon dioxide, and nitrogen gas, right? It can diffuse, it can dissolve, yeah, right, in the lipid bilayer. It can pass through the membrane rapidly, okay? Whereas there are other ions such as the chloride, yeah, sodium, potassium, okay, and also the small uncharged polar molecules such as the water and ethanol, and the large uncharged polar glucose such as the glucose and the sucrose. Okay, they cannot across the membrane easily. Yeah, they cannot across the membrane easily because of the hydrophobic core of the membrane. Okay, now we look at the transport protein where it allows the passage of hydrophilic substance across the membrane. So these channel proteins have a hydrophilic tunnel, okay, that certain molecules or ion can use as a tunnel, okay, and this channel protein we call it as aquaporins, okay, where it facilitates the passage of water, okay, only the water, okay, for aquaporin. The words aqua, okay, refers to water, aquapor. Porins come from the word pores, aquaporins. And then we have the carrier proteins, where it binds to a molecule and change the shape to shuttle them across the membrane. So this one, it makes the ATP, the energy. Okay, so a transport protein is a specific for a substance it moves. Okay, then we have this two types of transport across the plasma membrane. One is the passive transport, the other one is the active transport. Passive transport, it doesn't need any ATP, okay, or energy, whereas active, it needs the ATP. Okay, so passive transport can there are diffusion and the facilitated diffusion. Okay, so we will go for the different types of passive transport. Okay, the simple diffusion, for example, like the osmosis, and the facilitated diffusion. Okay, for simple diffusion, this molecule has tendency to spread out evenly into the available space. Okay, and the molecule will move randomly, but diffusion of a population of molecule may be directional. Okay, so molecules at dynamic equilibrium, as many molecules cross the membrane in one direction as in the other. So like the molecules, right, in given here, the example here, okay, you can see the dye here, the molecule of dye, it will cross through the membrane, okay, until it will reach at the equilibrium level. Yeah? So the molecules diffuse from more concentrated to less concentrated. So meaning that diffuse down its concentration gradient. So if it is down, then from concentrated to less concentrated, okay, and leads to dynamic equilibrium. The solid molecule continue to cross the membrane but at equal rates in both directions. Okay, now we look at this this one, the next one. Okay, a simple rule of diffusion. In the absence of other forces, okay, a substance will diffuse from where it more concentrated to less concentrated. Yeah, a substance will diffuse down concentration gradient. Okay, no energy needed and a spontaneous process. Okay, a diffusion of water across a selectively permeable membrane from the region of lower solid concentration to a region of higher solid concentration until the solid concentration of both sides of the membrane are equal is called as osmosis. Osmosis. So osmosis is the movement of water through a semi-permeable membrane, according to the concentration gradient of water across the membrane, which is inversely proportional to the concentration of solutes. While diffusion transports material across membrane and within cell, osmosis transports only the water. Water across a membrane, and the membrane limits the diffusion of solutes in the water. Okay, so you can see here, these are in the figures here, these are the green molecule here. The green color molecules is the sugar, right? And this part is the lower concentration of solute, and here is the higher concentration of solute. So the water will move through the permeable membrane, selective permeable membrane, okay, until it reach at the similar concentration of solute. Yeah. So these are the U tube glass with a selectively permeable membranes where it's separating the two sugar solutions. Yeah. So here water move from an area of higher to free water concentration, lower to higher solute concentrations. All right. Okay, so water molecule can pass through the pores, but sugar molecules cannot. Okay, only the water molecules. Okay, so this side has fewer solid molecules, more free water molecules. Okay, whereas in this side, the water molecule cluster, you can see here, it will cluster around the sugar molecules, and this side has more solid molecules, fewer free water molecules. So, and it has the selectively permeable membrane at the center here. Okay? So this the osmosis. Osmosis. Only the water molecules will move. Okay, now we look at the tonicity. Okay, so tonicity can be described how an extracellular solution can change the volume of a cell by affecting the osmosis, or tonicity is the ability of a solution to cause a cell to gain or lose water. Okay. There are three types of solution. One is the isotonic, the other one is hypertonic, and the third one is hypotonic. So if it is isotonic, the solid concentration is the same. Is that inside the cell, and there is no water movement across the plasma membrane. If it is hypertonic, the solid concentration is greater than inside the cells. The cell will loses the water. Hypotonic, solid concentration is less than that inside the cell. Okay, so the cell will gain more water. So we look at the blood cell here. Okay, when place the blood cells in a solution that is hypotonic to the cell, so what happens? The water will move faster. It will enter the cell. Yeah? Then it leaves the cells. Yeah? So later the cell will swell and lyse. It will burst, because the water move enter the cell faster. For the isotonic, if the if the blood cell is immersed in the environment of the isotonic, there is no net movement of water across the plasma membrane. So the water flows across the membrane at the same rate, both direction, in and out. Yes. However, when put it in the hypertonic, the cell will lose water to the environment. Okay, and the cell become shrivel. It will become shrivel, and then it will die. So shrivel here, it will contract and then it will die. Then this effect, example given here is the effect of increasing the salinity of a lake to the animal. Okay, now we look at what happens when we put the plant cell in place the plant cell in a hypotonic solution. So remember that the plant cell have the cell wall. So when put in the hypotonic solution, the water enters the cell by osmosis. The plant cell swells, but it does not burst. The cell become turgid, become very firm. And this is very important for the herbaceous plants. So, and then we look at the isotonic solutions. When we put a plant cell, there is no net movement of water molecules. no change in the cell volume, but the cell become flaccid, and then it will wilted, it will wilts. And for hypertonic solution, the cell will plasmolyze. There's plasmolysis. There's process of plasmolysis occur here. There's a contraction of the protoplast of because loss of the water, the protoplast of the plant cell as a result of loss of water from the cell. Okay. So there is a net outflow of the water by osmosis from the cell. Okay. The cell vacuole will shrink. Okay. As you know that plants have a large vacuole. Yeah. So if there more water flows out, then the plasma membrane will pull away from the cell wall, and the cells will shrivels. All right. Okay, the movement of solid across the membrane will, with the help of transport proteins, facilitated. This is about the facilitated diffusion. It follows the concentration gradients, increase the rate of diffusion across the cell membrane. Transport protein is very specific on which chemical they allow to pass through. Okay, so you can see here the transport proteins. Okay, there are channel here, right? Channel. The substance the solutes can move through the channels, proteins here, and the carrier protein here, right? It will change the shape of the carrier proteins. Okay, channel proteins passageway that allows specific molecule or ion to cross the membrane. Okay, and hydrophilic passageway that allows water molecule or small ions to flow very quickly from one side of the membrane to another. So example like the aquaporins or ion channels. Yeah. So these are the aquaporins. Okay, you can see here the water molecules. Okay, aquaporins are the water channel proteins. It facilitates the massive amounts of diffusion of water molecules. Yeah, in the plant and also in the animal cells. Okay, so if there is no aquaporin, the rate of water movement across the phospholipid bilayer is very slow. Okay. Now we look at the ion channels. It can be leaked channels or it can be gated channels. So what's the difference among these two? Between these two, leaked channels is always open. Yeah, it's always open. Whereas gated channels, it can open, it can close, okay, in response to a stimulus. The stimulus can be a chemical or ligand, yeah, or it can be electrical or change in voltage or mechanical. Okay. So now we look at the ion channels here, the figures, okay, where as I mentioned just now, non-gated or leaked channel always open. Okay, it's always open and specific for one type of ion, although not absolute. Gated ion channels, yeah, the ligand gated. It is an open or closed in response to ligand binding to receptor as acetylcholine. Okay, so the ligand gated ion channels, permeability is greatly increased when some type of chemical ligands band to the protein structure. Okay, so a large subset function as neurotransmitters. So acetylcholine is a neurotransmitter. Yeah, as neurotransmitter. Can see here the receptor. Yeah, the receptor here at the phospholipid. membrane, plasma membrane. Okay, so the receptor here, neurotransmitter, chemical, it will attach to receptor. Neurotransmitter chemical, it's like the acetylcholine, can attach to the bind site here, the sites, the sites here, and then it will open. Just now, if there is no transmitter chemical acceptor, it close. And the chloride, the potassium, and the sodium, can move in and out through these channels. So this is the chemically gated ion channel. So if you look the acetylcholine, the acetylcholine is an organic chemical that functions in brain and body of many types of animal, including humans, as a neurotransmitter, a chemical message released by the nerve cells to send signals to other cells such as neurons, muscle cells, and the gland cells. Neurotransmitter is the chemical that motor neurons of the nervous system release in order to reactivate or to activate the muscles. Okay, now we look at the voltage gated. This one, voltage gated, is open or closed in response to small voltage changes. So voltage gated ion channels, known as voltage dependent ion channel also, are channels whose permeability is influenced by the membrane potential. So you can see here the positive charge and the negative charge. There's a membrane voltage changes occur here, right? So here when the ions, when the ions cannot move in, but because the gate is closed, but if there is a voltage changes, so this one is positive outside here, and negative, but voltage change, the outside become negative, inside is positive, then the gate will open. This allow the ions to move in. So the voltage gated ion channel, you can see when it closed and when it's open. So it open or close in response to the small voltage changes. Okay, now we look at the channel protein example given here is the cystic fibrosis. This cystic fibrosis is a kind of genetic disease, where the mucus will block the airways. So it is characterized by the production of abnormally thick, sticky mucus. Mostly affectation. The mutation can cause defectivity of the chloride channel, and chloride transport across the membrane closely linked to the sodium transport. Cell lining the respiratory tract cannot absorb the sodium chloride. So the natrium chloride will accumulate, affecting natural defensive protein, which is called as defensive, that kill bacteria in the lung. So bacteria triggers airways or cell lining the respiratory tract to produce excess abnormal thick and sticky mucus. So there is a clogged airways. This will damage the lung, and there's a failure of respiratory respiration. Okay, so you can see here the mucus blocks the air sacs. These are the alveoli in the lungs. Okay, the mucus blocks the air sac, and also the mucus blocks the airways of the respiratory system. Okay, carrier proteins. Okay, now we look at the carrier proteins here. Some small hydrophilic organic molecules, example like the glucose and amino acid, use carrier protein to pass through the cell membrane. Okay, by facilitated diffusion. Through it change the shape. Okay, the change in shape can be triggered by the binding of the solute to the binding site. So these are the binding site of the carrier protein and release the transported molecule with no energy input is required. Okay, so these are the example. Okay, the glucose transporter, glucose bind to binding site of specific carrier protein, and the protein will change the shape, and then the glucose will be released into the cells. Okay, now we look at the example here. Okay, the carrier proteins. The carrier proteins, cystinuria. Cystinuria, it is an inherited disease. This is caused by too much of amino acid cysteine. Okay, also lysine or arginine in the urine. Normally, after entering the kidney, cysteine dissolves and goes back into the bloodstream. However, in people with cystinuria, carrier proteins that transport cysteine across the membrane of kidney cell are defective or missing. Okay? So there is no carrier protein. Yeah? So as a result, cysteine accumulates in the urine and form crystal or stones. So it will block the kidney ureters or bladders. So you can see here in the kidney here, can see stones. Stones. Okay, so the cysteine accumulates in the urine, forms crystal or stones. Okay, you can see here the stones at the bladder and also this part of the kidneys. Okay, so this is my last slide on the questions. Okay, so there are 3 questions. The first one is describe simple diffusion. Second one is what happened to an animal cell when immersed in the following solution in the hypotonic and the hypotonic. And then name 2 types of ion channels. Okay, this is the last slide. With this, I end my lectures. Thank you.
Okey, terima kasih banyak-banyak Dr. Dan juga terima kasih saya ucapkan kepada Profesor Madya Dr. Norrizah kerana sudi meluangkan masa bersama kami berkongsi ilmu dan pengalaman kepada pelajar sains asasi dengkil dan juga kami pensyarah biologi. Okey, semoga kita dapat berjumpa lagi di masa akan datang. Okay, thank you very much Doktor.
Okay, welcome. Jumpa lagi. Okay, terima kasih banyak-banyak.
Okey, kepada YouTube channel. Okey, di paparan chat kami menyediakan link untuk kehadiran hari ini dan juga sebentar lagi kami akan presentkan QR code. Okey, so boleh klik di mana-mana chat di YouTube